Hey team, Dr. Jack Gordy here, and in this video I'm going to be taking you through the immune system. It's just going to be an introduction to the immune system, but I'm super excited because I'm an immunologist, so this is my favorite thing on the planet. It's amazing. Anyway, let's jump into it. The immune system. So first of all, what is the immune system? We talk a lot about it. We hear a lot about it, boosting your immune system with vitamins and whatnot. So what actually is it? Well, it's all the processes, proteins, cells, and structures in an organism that help protect it from communicable and non-communicable diseases. So that's key there. It doesn't just fight pathogens like bacteria and viruses. It also helps us with things like cancer. Cool. So what's included in the immune system, if it's all the structures and everything? We typically break it up into three main groups. So there are surface barriers. There's the innate immune system, and within the innate immune system, there's what's called the humoral immunity and the cellular immunity, and I'm going to jump into that a little bit more later on in the video. Then there is the adaptive immune system, and that too has humoral immunity as well as cellular immunity going on. Now, what do we mean by this? Well, imagine taking your blood and then letting the cells settle, taking the liquid out of the top, and then taking the cells, right? Well, what it turned out when they were doing early research is the liquid, the humor, if you will, which is what Greeks call the fluids of the body. So the humor of the blood was able to kill pathogens and bacteria, for example. But so were the cells. So they divided the immune system up in that way. There's humoral immunity, which is the liquids of your body and the ability of that liquid to kill pathogens. And then there's cellular immunity, which is the ability of the cells to kill pathogens too. Now, I've got this lovely little picture of a tank here. And then down here, I've got the CIA. Well, that's just a really good analogy for the two different kinds of immune systems. They're both effective at killing pathogens, the enemy, if you will, disabling and getting rid of the enemy, if you will. But the innate immune system is often associated with doing a bit more collateral damage. It's like a tank. It's going to blow up the area, which is going to do damage to you as well as the pathogen. And they kind of, if you're a tank, you've only got one option, which is shoot your shells, right? You blow it up. You've only really got one approach to getting rid of the pathogen. The CIA does investigations. It learns who the bad guys are. It remembers them, and it attacks in a specific and silent and deadly way. That's a really good way to think about the two immune systems. And as I break it down, you're going to see why that's the case. But first, let's look at surface barriers. A little bit boring, I'm not going to lie. It gets way more interesting when we start talking about cells and humors. So yeah, the skin, not very exciting, but the skin has, for example, a robust keratin outer layer, which is actually dead. The outer layer of your skin is kind of dead, which helps protect us from all sorts of pathogens, right? Because it's sort of dry and dead and not really a great place for life. It helps prevent pathogens getting into the moist and delicious body inside. Now, although I was ragging on the skin and surface barriers, they probably protect us from more pathogens than all the rest of the immune system combined, right? And this is why when we get a wound, we've got to sterilize it because we've broken that barrier and now we might get an infection. Another one is tears and snot, and we will remember this from my video on Alexander Fleming. Before he discovered penicillin, he discovered lysozyme, which was an enzyme in his tears and his snot, which just has the ability to kill bacteria, and it's just an enzyme that can lyse bacteria. So a lot of our surfaces have antimicrobial proteins and compounds in them to help protect us from any invading pathogens, because they're constantly exposed to pathogens because they're exposed to the outer environment. And lastly, we've got stomach acids. Now, you often think about stomach acids for digestion, but there is actually a major reason. One of the major reasons why we have an acidic stomach is to help sterilize it so we don't get too many gastrointestinal infections or helminth parasites or anything like that. The acids help break down and destroy these pathogens as they come through the stomach. Okay, so now onto the innate immune system. Now, one of the key things, you know how I was talking about, it's kind of crude. The innate immune system has a playbook, it has an instruction manual, it has an approach for pathogens, but it's not very specific. It's sort of a general response to viruses, a general response to bacteria, a general response to fungi, and a general response to parasites. You could almost say the playbook only really has 4 plays, 4 instruction manuals for anything. So if you've got influenza or SARS-CoV-2, your innate immune system is going to undergo very similar processes to deal with that disease. So it's not a specific approach, and because of that lack of specificity, it can cause damage to your surrounding tissues. And you'll see why in a second. Now, again, this can be broken into humoral and cellular immunity. So let's just have a quick look at humoral immunity. Let's say there's a pathogen in your blood. There is a whole series of proteins called complement proteins in your blood. Now, these complement proteins, all by themselves, can recognize some pathogens. So they recognize the pathogens and they become activated. And once they become activated, we get what's called the complement protein cascade. And you're going to see this kind of exponential growth graph a lot when we talk about the immune system. The immune system wants to take a very small signal, maybe it's only detected 1 or 2 pathogens, and has a large response. So we need to amplify that signal, right? So one complement protein will activate several other complement proteins, which will activate even more other complement proteins, and we'll get this exponential growth in activation. So we'll get some sort of cascade like this until we get to the final complement proteins. Now they can assemble into some really interesting structures. So this is a little donut with a hole in the middle of it. And the complement proteins have formed a donut. What is that donut going to do? Well, it turns out the chemical composition of that donut allows it to insert itself into membranes. So we call this the membrane attack complex, and it's going to insert itself into membranes. So here we can actually see it under the electron microscope. It has inserted itself into the membrane of a bacteria. And what it does, because it's donut shaped, it's created a hole in the membrane of that bacteria, and it's going to cause them to pop and lyse. Now that's kind of cool, right? We've got just these proteins, no cells, no thinking thing involved here that's just floating around our blood. And it's just going to recognize, activate, and kill pathogens all by itself. That's pretty magical, and that's pretty awesome. But I gotta say, I'm more of a fan of cellular immunity. So the innate immune system also has a cellular component, and this cellular component has 2 main weapons that it kind of utilizes for everything. One is phagocytosis, and the other one is toxic compounds. So phagocytosis is eating. That's cell eating, right? If you think about your esophagus, that word phag, P-H-A-G, means eating. So phagocytosis is the eating of cells, or cellular eating. So what happens here is we've got some viral particles outside the macrophage or a phagocyte. A phagocyte is a cell that eats. It's an innate immune cell that eats. So a phago means eat, cyte means cell. So a phagocyte is a cell that undergoes eating. So we've got a phagocyte here, and what it's done is this membrane has enveloped some viruses and budded inwards. That creates what's called a phagosome, full of the pathogens, and then the lysosome, which is an organelle inside all cells, is going to fuse with the phagosome. Now the lysosome is the stomach of the cell. It's really acidic and it's full of digestive enzymes. So when it fuses with the phagosome, it forms what's called a phagolysosome, and it destroys whatever pathogens inside there, right? So this is one of the major weapons of the innate immune system, to just start gobbling up things in the extracellular fluid. The other one is toxic compounds. Now, there is a huge list of toxic compounds, but one of my favorite is innate immune cells actually have an enzyme, which is a protein machine that facilitates chemical reactions, and this enzyme produces bleach. Like literally bleach. Your body produces bleach. And it does so for the exact same reason why we use bleach for cleaning sinks and everything like that. Bleach is a very reactive chemical that reacts and destroys whatever it reacts with, essentially, because the chemical composition of anything dictates its structure, which dictates its function. So if you have a chemical that reacts with everything, it's going to change the chemical formula of, say, a membrane protein of a virus or a membrane protein of a bacteria. It's going to change the chemical formula and therefore the structure and therefore the function of that, and it will destroy it. If it reacts with phospholipids, for example, suddenly the membrane stops working and you end up with lysis of the cell. So your body produces bleach, which, for the same reason we produce bleach, we use bleach in the bathroom to sterilize the area. But this is where you can see the non-specific nature of the innate immune system. It's not specific to whatever pathogen it's fighting. So that bleach is going to go and it's going to kill some of our host cells too. So this is where the innate immune system causes that collateral damage. And that's why I've drawn it with like a little tank there. When you're a tank, there's only one response, and that's to shoot your explosive out and kill sort of everything in the general area. That's the innate immune response. So let's have a look at some images here. Here, this is an innate immune cell we will meet later. It's called a neutrophil. It is a phagocyte. So it is hunting down this bacteria, and it's doing positive chemotaxis, right? So it is moving towards a upper chemical gradient. So this bacteria will be releasing a chemical, and the concentration gradient will guide the neutrophil up to attack this bacteria, and it then phagocytoses it, and it's now inside the cell getting digested, which is super cool. Honestly, it's almost exactly analogous to a sniffer dog, right? It's following the chemical trail of the bacteria up the concentration gradient, right? Yeah, so that's phagocytosis. Toxic compounds. Well, here we have a worm. Now, you can't phagocytose a helminth parasite. You can't phagocytose a worm. They're too big. So what are you going to do? You're going to swarm it and spray toxic compounds all over it. So here we have innate immune cells. We're going to meet these guys later, but they're called eosinophils, and they are absolutely swarming that parasite, spraying it with toxic compounds, and it doesn't stand a chance. It's all its cells will be lithing under that toxic soup of chemicals that are being released by those immune cells, and it is dead. It's 100% dead. That one there. So there's the innate immune system working incredibly effective. What about the adaptive immune system? Well, I mentioned it's like the CIA, and it's like it's got a case file, not just for each disease, but often each variant of the disease. And it learns exactly how to attack it in a very specific way. So it uses, when it attacks and kills a pathogen, it does it quietly, and it does it directly, and it does it specifically, which is the complete opposite of the innate immune system. It also has the ability to learn. So when you take a vaccine, for example, your body is learning what that pathogen looks like, so it can make a better response the next time it's exposed. That's your adaptive immune system. So by injecting you with a vaccine, which might be a dead virus, for example, your body is creating this file. It's creating its little dossier about that one pathogen. Now, the advantages is it's quiet, and so it doesn't cause collateral damage, and it's specific. That's the advantage. The disadvantage is that it's a bit slow, and it's so specific that sometimes that hurts it, right? So this is why we have to get an influenza vaccine every year. It's because the virus is constantly mutating. So the file is so specific that it doesn't recognize the next flu virus that it sees in the next season, because it's mutated and changed ever so slightly. So there are some advantages and disadvantages to the adaptive immune system. So how does it do it? Well, it has two main weapons. One is the antibody. I've drawn the logo as a little bit of glue, because you'll see that's pretty much how antibodies work. They're very sticky and gluey proteins. And the other one is celllysis. You might be like, why do you want to lyse a cell? Well, we'll jump into that. So first, let's start with antibodies. Antibodies are part of the humoral immunity. So they have the ability to kill and disable pathogens, and they're just a protein floating around in the fluids of your blood. And this is what they look like. They're often drawn like a little capital Y, and that's because the protein structure looks quite a bit like a capital Y. Yeah, so it's a secreted protein. It's made by the B cell, and I'm going to jump into that a bit later, but a B cell is just an immune cell that goes around your blood, and it also lives in your lymph nodes, those bumps under your arms. It's a long-lived protein, so a lot of proteins don't last that long in your blood. Some cytokines, which we'll learn about later, they last about 2-3 hours in the blood before half of them are gone. In this one, half of your antibody will break down and be destroyed after 1 month. So it's a very long-lived protein in the blood. And this is where the specificity comes in. Each antibody type, each antibody that's produced by a B cell is unique and binds strongly with one specific biological molecule called an antigen. Now they're called an antigen because they're an antibody generator, right? They're an antibody generator. If you inject an antigen into something, it will create antibodies for that antigen. So each B cell typically only produces one kind of antibody, and that kind of antibody that specific type of antibody that's unique to that one B cell will bind to one pathogen, which is crazy. It's crazy. So let's have a look at an example of an antigen, an antibody generator. Here's the coronavirus, SARS-CoV-2, the one and only. And on the surface of it, you can see this little spike protein. We've heard a lot about spike proteins, but there it is, viewed under an electron microscope. Here we were able to construct the molecular structure of it, and this is what it looks like. If we were to inject that into someone, which actually one of the vaccines is just injecting the spike protein, I think it's Novavax, we would generate antibodies that would bind specifically to that spike protein. So what's so bad about being bind to? Well, what do antibodies actually do when they're binding to it? Well, they do a couple of things. They do quite a few things, but one of the main things is neutralization. So if you think about the SARS-CoV-2 virus, to get its genome into your cells, what it does is it has its spike protein which binds to a receptor on your cell surface. So that spike protein binds to a receptor, it then pulls its genome into your cell. So that's how it gets its genome into your cell. But if you cover those spike proteins in antibodies, so look at this coronavirus, it's covered in antibodies that have all bound to the spike protein. This is how we draw antibodies with that capital Y structure. Then the virus can't get into the cell. It's lost its spike protein, which it needs. It's lost the binding domain. It can't latch on. We can't get an interaction between that spike protein and the cell receptor. So this is called neutralization. Essentially, coating all the virulence factors of any pathogen prevents it from infecting you, right? So antibodies can bind to virulence factors, and if they bind to virulence factors, they prevent the pathogen from being virulent, from being infective, right? So you can block it by blocking the active compounds on the surface of any pathogen. Another one is agglutination. So there's two binding domains. That's why it's a capital Y, right? It's a capital Y. It's got one binding domain over here and one binding domain over there. By having two binding domains, it means you can bind two pathogens. And so here we can see the capital Ys, each of them are binding a couple of SARS-CoV-2s, and that kind of sticks the pathogens together, and it precipitates them so they're now not floating around the fluid. It makes them very easy to phagocytose with the innate immune system coming in and just gobbling up these chunks of pathogens, and it prevents the pathogens from flowing all throughout the body, right? It clumps them together, disabling them, neutralizing them, gluing them together, agglutination, gluing them together, preventing them from being pathogens. So what was the other one? The other cellular approach was celllysis. Why would you want to lyse a cell? Well, antibodies can't get into our cells. They just float around the fluid, the extracellular fluid that our cells are bathed in, or our blood. They can't get into a cell. But in cells is where a lot of pathogens are. Viruses are in there, but also intracellular bacteria like chlamydia are inside your cells. So what are we going to do? Are we just going to let that cell keep producing that pathogen? Or are we going to go and lyse that cell? So we've mentioned the B cell. The B cell produces antibodies. Now the other main adaptive immune cell is the T cell. So what all cells in your body are doing, is they reach inside their cytosol, the fluid inside the cell, they grab some proteins and they display it on the surface of the cell. Cells are always displaying this. Now we call this antigen presentation, because remember, antigens are just those fragments of pathogens that often generate antibodies. So you can present an antigen from inside your cell, outside of your cell. Now, normally it's just your own protein. So you say, hey, look, I'm just full of my own proteins, and your immune system can come around and check to see, is that protein a self protein or is it something different? So here we've got a lung epithelial cell, and it is infected with SARS-CoV-2, with a virus, right? So now there are viral proteins inside that cell. So it does what it always does, what all cells in your body do. It grabs some proteins, displays it on its surface, but this time it's not a self protein, it's a viral protein it's displaying. So here we've got a spike protein that is now being displayed by the lung epithelial cell. Along will come a T cell. Now, a T cell will have a T cell receptor. Now, a lot like antibodies, each B cell produces one antibody type, and that one antibody subtype, and that antibody can only recognize one antigen, right? So there's in your body right now, there is a B cell that produces a antibody that can bind to a fragment of the spike protein, right, from SARS-CoV-2. In a very similar way, T cells have a T cell receptor, and that T cell receptor only recognizes one antigen, one pathogenic biological molecule. Now, this biological molecule could be a carbohydrate, it could be a protein, it could be a lot of things. It's often a protein. So this T cell is the only cell that can recognize that exact fragment of that viral protein in your body. So it's come along, it's found it, and it's recognized it, and it goes, that is not a self antigen. So T cells get trained to not recognize your own molecules and only recognize one pathogenic molecule. So this T cell has now recognized this spike protein using its T cell receptor, which is unique to this T cell. And it will now release cytotoxic and apoptotic inducing compounds, right? So we remember apoptosis, programmed cell death. So it is now going to tell that lung epithelial cell, you need to die because you're producing viruses. You are just a harbinger of pathogens, so we need to kill you, right? So that cell will then undergo apoptosis and safely die and stop producing the viruses. So that's a really important part. So the T cells are attacking cells and killing any cells, any of your host cells that are producing viruses, and your B cells are producing antibodies. Now, this one T cell isn't going to be the lone guy fighting this virus. She will now divide and divide and divide and divide. So now you've got lots of this T cell with that exact T cell receptor. And it's the same with the B cell. The B cell will divide and divide and divide. So now we have lots of B cells producing the right antibody to fight the pathogen. Right, so that was a brief intro into the immune system, and I'm going to dive much deeper on that in the next video, in the next couple of videos. Up next, we're going to have a deeper dive into the innate immune system. We're going to look at some of the cells in there, and then we're going to jump into the adaptive immune system. Yeah, brilliant. So I'll catch you in the next video, guys.